Welcome to our world.
This is your global.
Invited me to this meeting, and I have no conflicts of interest for the moment. I have had before grants from Chiesi Pharmaceuticals, but not the last 4 years. And I shall talk about surfactant. As we already know, we heard it many times today and yesterday, the birth every year, the total birth is at 130, 140 million babies a year. 1/10 of them are premature, 10 to 50 million, and 1/10 of that have RDS, respiratory distress syndrome. And it's especially the very premature babies that have immature lungs. And I have worked with immature lungs and lung surfactant for more than 40 years. And I think the interest for immature lungs began in the beginning of the 90s, but especially it was interesting when President Kennedy got a preterm baby. Not so preterm in our days. It was born in about week 34, 35, nearly 2 kilograms of weight, but died within 2 days in 1963. And why is surfactant important? We know today we must have air, we must have oxygen to live, to breathe. We must inspiration, oxygen, and expiration, carbon dioxide. And these premature babies have a lack of surfactant and they cannot breathe. Why not? They cannot open their alveoli, and the alveoli are filled with liquid. So what happens? In a grown-up people, we have perhaps 400, 500 million alveoli. In a newborn baby, not preterm, we have about 1/3 of that. And we can see the area is very important too. The area is about 1 square meter per kilogram body weight. So through this air, we should take up oxygen. What had happened with surfactant research? In the beginning of the 19th, a man called Professor Nergård wrote about some substance in the alveoli that was important. And what was that? And it was clarified by John Clemens in the 50s, and in the 60s and said, in the alveoli we have phospholipids. And phospholipids are very important to change the surface tension, to diminish the surface tension, and also to prevent the alveoli to collapse during expiration. And also in America, there was a doctor called Mary Avery. She was also very important. She saw a combination between low surfactant and RDS, hyaline membrane diseases it was called at that time. And she was a long time also at the Boston hospital. And in Boston Hospital, we also had the baby John Clemens' child, who didn't survive in the 1963. But what is important here in the alveoli, we have a fluid surface, we have a monolayer of phospholipids. And John Clemens said already in the 60s that this is the d-palmitoyl phosphatidylcholine. That is very important, so diminish the surface tension during expiration. And phospholipids have a general formula of a glycerol skeleton, 4 fatty acids, a phosphate group, and then can have the colon, serine, or some sort. X can be different types. But it was the d-palmitoyl phosphatidylcholine that was important for lowering the surface tension in the alveoli. But what they do, as usual, they made a clinical trial. They made a clinical trial with d-palmitoyl phosphatidylcholine, nebulized. What happened? No clinical effects. And they wrote a paper. The underlying cause of RDS was no pulmonary blood flow rather than primary surfactant deficiency. They thought at that time. But you know, it's very important that the material you give, the surfactant, is spreading in seconds over the big surface. And if you have a surface that is 1 square meter per kilogram body weight, if you have a fat, it will not spread like that. That was the reason it didn't function at all. So, if you have a project, and that to me and all other people, I think it's important that you have good collaborators. That's the most important thing. You have to have very good collaboration. You can't do anything alone. You must collaborate, and you must have good luck. And it was so, when I started my research in the 67, I started by chance with phospholipids. And we found some activity there, and my problem was to isolate phospholipids, separate them, and my thesis was about phospholipids and the turnover of phospholipids in animals. But what should I do with all the phospholipids? What is interesting? Now today we know it is very interesting, and in all membranes we have phospholipids. In 1980, a man called Bengt Robertson came to my lab and asked about, he said, I'm working with surfactant. And I take alveolar wash from adult rabbits. And then I centrifuge it, take the rest of it, suspend it in saline, and sometimes it functions very well, sometimes not at all. What is the wrong with the phospholipid composition? And I said, the way you do it, you will destroy phospholipids. You heated it to 90 degrees. Sometimes you will destroy 5%, some 50% perhaps. And I told the bank that perhaps we'd make a collaboration. I make the surfactant with my methods, and you have animal experiments, and you use those experiments. For it was a pathology. We were both laboratory doctors. So he showed me some results, and he said that you can see here, when I give surfactant, most of the preterm babies will survive, but with saline or none, they die within a quarter of an hour. So we started our research together in 1980, and we took my methods for isolating phospholipids, his methods for in vitro and in vivo tests. And we started with pig lungs, and you can say, why not cow lungs? Yeah, it's the same. We have made it with that too. But pig lungs could be got free from the slaughterhouse when we had to pay for the cow lungs rather much. And we had our grants. So, of course, we took pig lungs. And we minced them. We took them in normal cell lines, shake them, filtration, centrifugation, and then we had the raw material. Extracted organic solvents and got the lipid extracts. And I think many have stopped there with lipid extracts, but we said it's very important to also to get the phospholipid fraction, to get the real active substance, not to have all neutral lipids with it. So as you can see, we separated the lipids in a phospholipid, took the phospholipid fraction. We have free fatty acid cholesterol 3-acylglycerols, and cholesterol esters, and they were discarded. Also to get the phospholipid fraction. And we took that phospholipid fraction and gave it to the premature rabbits, and it functioned very well. And we repeated day after day. I think we had about 80 to 100 adult rabbits every year, and each of them had a mean value of 6 premature babies. So we made rather many animal experiments in the beginning of the 80s. And as you say, importance factors for the surfactant, they are rapid absorption, and it is spread over a large area, and they said reduce surface tension, give an alveolar stability, and it must be rather resistance to inactivation. Then came the question, how much should we do? And that was in our animal experiments in the beginning of the 80s. How much shall we give? We calculated from the literature and said 100 to 200 milligram per kilogram body weight. And we tested both of that. What concentration? So I think we should have a high concentration. Both of us thought the same. A high concentration, and we started with 100 milligram per mil. The viscosity was so high. We lowered it to 50, okay. And then we take it between, perhaps 80 milligram per mil. And after we had tested a couple of months, perhaps a little more, 200 milligram per kilogram body weight and 80 milligram per mil. And I think the company still uses that concentration and that amount as a standard dose. So when we worked with all this, we had also an animal model. And we had rabbits, we had preterm rabbits. It's 27 days, and 27 days they were very preterm, and lack of surfactant. And we ventilated them, and we measured tidal volumes, and we also measured lung gas volume at end expiration. It was very important that the lungs were not, alveoli were not collapsed during end expiration. So here is only a model that we have animal preterm rabbits. The weight was about 20 to 25 grams of each rabbit, and we ventilated them in parallel and made this type of experiments. So when we measure the tidal volumes, you can see here in the VT the tidal volume, and it was very important that we had a rather high lung gas volume at end expiration, that there were not collapsed alveoli during expiration. And we can see in the next We had three, surfactant 1 and surfactant 2 and non-treated. The non-treated, low volumes, tidal volumes, lung gas volume was very low. And then we say surfactant 1 and surfactant 2, about the same tidal volumes, but lung gas volumes was different, 1.4 to 12. In the first surfactant, the alveoli were collapsed during end expiration, but not in the second. The second surfactant was a good surfactant for us, not the first one. So it's very important that the surfactant will be reduced the surface tension during breathing, and that we have a rather high lung gas volume. So the conclusion was, as I said before, measure both tidal volume and lung gas volumes, and we measured both of them. On both was very important. And we made many experiments every year, every day. And in 1983, they called us from the hospital. The doctor there, responsible doctor, the professor there, called us and said, I know you are working with surfactant. I have a child born in week 27, 790 grams. He will die within a couple of hours. We have nothing more we can do. You have made written consent from me and also from the parents. Can you give any surfactant? I mean, of course, in 1983, both Bengt and us were a little afraid. Can we do that? Can we give it surfactant, this type of surfactant? But we gave it surfactant. And you see, this is the suspension we gave, 80 milligram per mil. And we thought also it's very important to have liquid-filled lungs. You shall give a low volume and high concentration of surfactant. So therefore I think it was very important that we could give a concentration of 80 milligram per mil, and not 25 or 30, and large volume. So the first baby, I think this was the second baby, we had no pictures from the first baby. It was so exalted at that time. And they happened the same with the first, and then the second time we gave surfactant. The values about in the ventilator, FiO2 was about 85%, and within an hour they could breathe air. And the blue baby became pink. So that was our first experiments, and we treated perhaps 10 babies before we started the clinical trial for vital indications. But that was the surfactant GNSA, Gris natursurfactant, and in English, porcine-natural surfactant, translated. And what was that for type of surfactant? And Bengt and I should have lunch after an animal experiment, and we said we must give it a name. We must give it another name. And then said we take the two first letters from my name and the 2 letters from your name, Curosurf. And I think that in most countries they use Curosurf as a name today. We gave it a name during a lunch, a cheap lunch after an animal experiment, and I think that is the name used in most countries. I think in nearly all countries without 1 or 2, they use the name Curosurf. So, another thing, if we make this surfactant for an animal experiment, from one lung, pig lung, you could get 2 or 3 vials, to treat 2 or 3 babies with surfactant, not more than that. And of course, we showed the clinical trial. We made the clinical trials. I got the surfactant from the slaughterhouse in Uppsala and in Stockholm. And we had about, in each batch, about 50 to 100 gram of lungs in our hospital laboratory. So that was rather a large amount, and not so much surfactant anyway. And what was this surfactant? Yes, the composition of Curosurf should be only phospholipids. And because I'm a clinical chemist, I was interested in looking at the phospholipid composition, no problem. I had it done for many years. But I also found a small amount of proteins. And the proteins, the peppers, they were very important. And that was, as you know, no SPB and SPC. And what should I do? Was it contaminants, or what was these proteins? So I talked to, I know him very well, the best protein chemist in Sweden at that time, Hans Jörnvall, professor at Karolinska Institutet, and I told him, I'm doing so I'm purifying that or that way. And he said, it must be contaminants. It must be contaminants. But I shall make, we knew each other very well, and I shall make an experiment to see. And then he called me after a couple of days and said, Tore, it's not 50, it's only 2. If you can separate them from the phospholipids and separate the peptides too. I said my best student, Jan Johansson, now a professor at Karolinska Institutet, and to look at the sequence of these peptides. And Jan Johansson and I, we have worked together until I retired a couple of years ago. So that has been very successful, and I think collaborators are very important, and also good luck. So why do we need these peptides? They are hydrophobic, yes, for spreading, formation of multi-layers. And we tried all, and we could also at that time in the beginning, separate the phospholipids from the peptides. And if we only took the phospholipids, it didn't function. But if we added these 1 to 2% of proteins, it functioned very well. So the proteins, they were very important. So what happened after that? We thought, Bengt and I, that we had a very good product. We must have a clinical network. Both Bengt and I, we were not neonatologists, we were laboratory doctors. We must try clinical trials. We must have a network of neonatologists to succeed with that. And we must get a pharmaceutical partner. And we must start with research meets, perhaps every year. And important for all this was that, first of all, Professor Henry Halliday from Belfast visited us, and he was very important for this surfactant research for us. Jag, Bengt, and Henry, we had a dinner in 1984, I think, in November 1984, and they called us from the hospital during our dinner and said, we have another child, can you give any surfactant? And Henry said, I will be there, can I accompany you to the hospital? Yes, of course. And the same thing happened. The blue baby became pink, and they could reduce the ventilator settings with oxygen. So that was very bad. And the day after, he should go back to Belfast in Northern Ireland, and he told, I would be a member of this clinical trial? Can I? Of course you can. That was November. And you can have 10 vials of it. At that time we had it freeze dried, and he took it in the hand luggage in the plane, without any declaration, I think we had at that time. And I think today we couldn't have done it, but this was in the 80s. So the second one who visited Professor Christian Speer from Göttingen at that time it was, and he was also a couple of weeks with us, and we ran some experiments. And both those people, both Henry Halliday and Christian Speer, they were very important for our clinical trials when we started them. So we should make a clinical trial with the sickest baby. It was approved, and it started in '85 in January about there, and we should treat 140 babies and 140 about controls, and an interim analysis after half. This was an interim analysis, and after the interim analysis, they said you must stop the trial. You can't have a placebo group. You must treat all babies. And then we treated the next new series on the same indication. We treated all babies. And I think this is the only clinical trial with a control group. And we can see the mortality in the control group was 51%. In the first treat, it's 31. We learn more about surfactant. In the new series with the same indication, it was down to 15%. So it's very promising. After this trial, and also at the end of it, we started many other trials. And you can see these are the trials made with Curosurf under about 10 years. And the first trials, perhaps to 93, it was produced at the Karolinska University Hospital, and the rest from KC Pharmaceutici. So because it's very important that these big companies, such as in Sweden we had Astra and we had the pharmacy, they were not interested. They said it's too small product. It's too small product for us, and the marketing costs are huge. So it's not so interesting. So Bengt Robertson, Julio Bevilacqua at Parma, and he knew KC Pharmaceutici, the two brothers, and said perhaps they are interested. So Bengt and I visited KC, and they were interested in starting to producing this surfactant. And I think there will be good luck for them too. But here we have when we started, but also we can say it's very important when you have not change a method, because they had a subcontractor, and the subcontractor said, I do as you have said, but your method with the phospholipids I have to try to improve, so I don't take the first part of the phospholipid fraction, or not the second, only the middle one. And I said, you can't do that. Why not? Because the different types of phospholipids and the proteins in the first part of the phospholipid fraction. And if you do like that, you will lose the proteins, and then it will not function. We change it back. I talked to Paolo Chesi and the subcontractor, and they changed it. And they done it. And that was, it hadn't functioned. And then Chesa told me, perhaps it functions in laboratory, but not in industrial scale. But that was Paolo Chesi who told me, go to the subcontractor and see that everything is correct. So it was good luck. It's not by only collaboration, you must have a good luck too. After that, from '86, we have meetings every year. I think we have had about 40 meetings, research meetings. In the beginning, it was only research, one or several doses. When shall we give surfactant? How shall we give surfactant? And so on. But of course, it has changed a little. This is Henry Halliday to the left. And the second is a young boy, and Henry Halliday left our lab with the surfactant in November, and he treated the first boy in February. And this boy, also vital, is in the clinical trial. And this boy has now a family. He lives in US with family and children. So that is what happened. And what happened with the first boy we treated in June 1983? I got an email at Karolinska. I was first. Was it a spam or not? I thought it was a spam. The first, and then I said, I must look at it. And in this mail, I could see I was the first person who got Curosurf. And I checked the name and the parents and everything, and it was okay. And then we invited this boy, Patrick Svensson, and his parents to our house, and I was nervous at that time. What will happen with that boy? Will I have a boy, a blind boy? Will I have a boy with extra oxygen? Can he walk? What has happened with him? And then I had met a healthy boy, a healthy man. He was 33 years at that time. So that has been very interesting also. What happened with Curosurf? Yes, I think a couple of years ago, more than 7 million babies were treated with the surfactant, and more than 1 million lives saved. I think today 8-9 million, and perhaps more than 1.5 million lives saved. And it was launched in Europe 92-93. It's available in about 100 countries. But it's also a problem. It's a limited supply. And I think synthetic surfactant is needed. And I should take some word about that. First of all, this is person very important have been for surfactant. There were me and Bengt Robertson. Casey without Casey, it had been a problem. Ola Sagstad, who is here today, Henry Halliday, and Christian Speer. But you can make an animal-derived preparation, but you need many, many pigs. And I made a calculation that you can treat perhaps 30 to 40% of all premature babies with RDS. You have no preparation available for other lung diseases. You can have surfactant as a carrier of other drugs, for example. We have not enough. Time consuming to produce. Oh, it's rather expensive. And I think that synthetic surfactants are needed. Composition of natural surfactants is very complex. There are many phospholipids, perhaps about 50. There are 4 proteins, water-soluble and hydrophobic proteins. You don't need perhaps that complex phospholipid mixture, or you don't need hydrophilic proteins. But you had to try to make a synthetic surfactant. You had to reduce the number of phospholipids, different types, and you had problems with hydrophobic peptides. They are not stable when you try to purify them, and then you synthesize them. They are the right sequence of amino acids, but not the right secondary and tertiary structure, so you must modify it in some way. So what we did, that was, we took a mix of phospholipids, 2 phospholipids. We produced an SPB and SPC analog. We changed some of the amino acids to get a stable secondary structure. And it took us 5 to 10 years. Everything takes time. And we made together with Curstedt the synthetic surfactant 5633, and this was developed by Curstedt. The peptides were developed by us at Karolinska. And you can see we have a phospholipid structure, 2 phospholipids, ratio 1:1, and we have SPB and SPC analog. I made a clinical trial in the US. This is and the treatment with CHF 5633 showed similar efficacy and safety as Curosurf. After that, Curstedt has stopped this synthetic surfactant because they make rather much money of Curosurf. They don't need it for the moment. I think for all these preterm babies, we need a synthetic surfactant. This is not the ideal surfactant, CHF, but because the SPB is too low amount, or SPB, the analog, was very expensive to produce. So Jan Johansson and myself at Karolinska Institute, we made a combo peptide. We used a linker between the 2 SPB and SPC and made it recombinant. And it took us some time, but this new peptide, we combine SPB and SPC with a simple phospholipid mixture. It functions very well in premature animals and also in adult animal model. And we can produce it. We increased it in a bioreactor, so we can produce from 1 liter. Can we treat, perhaps we could treat about 50 babies, and it's much easier to have considered 75 kilogram of pig lungs. So I think in the future, I hope Curstedt will go on with producing these synthetic surfactants, and I've spoken to them about that also. And after that, I have retired because they are not working anymore in the lab, but we have some facilities left. So that's all I have to say. Thank you very much.
Thank you so much for an amazing talk, really going through the history. And to be honest, I did not know many gaps. I had a lot of gaps in my knowledge, so thank you so much for putting it all together. Really, really. We are so lucky to hear it from you. Any comments, any questions?
Yeah, thank you so much for that great presentation and all the things you have done to the community of neonatology and saving so many lives. Yeah, I mean, I'm inspired by the story, especially me being a surfactant RDS researcher. So really excited to hear about your work and what you had done to the community. I had a quick question about the original clinical trial, the multi-center trial. What was the gestational age in average? And because it was an up-and-coming treatment, did you have sort of pushback or difficulties enrolling patients who might be getting placebo? And how did that trial kind of worked out?
Perhaps you shall not ask me about the clinical trials, because I was at the laboratory, we made the surfactant. We have also this Henry Halliday, who died a couple of years ago, and Christian Speer, who was, they were responsible for all the clinical trials. And
Any comments, questions?
That was amazing. Now I can tell everybody why Curosurf is called Curosurf. I just thought Casey came up with that name. But I guess my question for you is that the surfactant production and the trials that were done were before the antenatal steroids came in the picture. And now there are certain babies that we still find are not responsive as well to the surfactant, right? We call them surfactant failures, even though we keep on doing the first dose, second dose, third dose. You know, we sometimes say leave well alone, but the trials that were done were on a specific patient population prior to the antenatal steroid era, and the dose is still the same, whether we are using it for that 22-weeker with RDS or that MECASP kid who's a 4-kilo child. So I guess my question is it time in future to start thinking of better identification of babies with RDS? Because those failures, are they because they just haven't cleared up lung fluid and can make endogenous surfactant? And then the dosing. Most of the neonatal medications, if you look at it, are tiered for gestational age at this point in time. So should there be room to start looking for alternative dosing strategies? And lastly, maybe tying in some more diagnostic therapies, because RDS is an X-ray. Yeah, it looks schmoozy, it looks hazy, let's give surfactant. But maybe integrating point-of-care ultrasounds to see whether we have parenchymal lung disease or we really have fluid sitting there, wherein the fluid babies will probably be non-responders and the one with true surfactant deficiency might do better. So just curious to hear what your thoughts on that are for future.
Of course, I think about different doses. It's okay. Nebulizing it also, and try different types of giving surfactant. So it's much more to do, and we need much more surfactant for doing all these type of experiments too. So I agree with you.
I have a question. I'm a big fan of your original thinking. And since we sort of met, I've been reading a lot about surfactant. But I think of myself more in the realm of immunology. And then I realized that surfactant protein A, B, C, and D are encoded in aquatic animals also. But surfactant came in when air-breathing animals came in. So why so? Why did these proteins get mixed up with phospholipids?
I don't know. I don't know why. Until the 80s, that you needed proteins. So I don't know. I don't know why it is so. But I have tried to purify different heart, muscle, and so on in the same way, and see if I have any hydrophobic proteins there. But I don't have. But I have it in the lung. And I don't know why. No, I can't answer that question directly.
Thank you. Yeah. I'll read and get back to you.
Yeah. I do think that these proteins are a part of the immune system, right? Kill surfactant B and C.
Thank you for that excellent presentation, Dr. Curstedt. You're talking about the composition of surfactant and the phospholipids and the proteins. But there are so many other components in the surfactant that's extracted from the lungs.
Yes.
Did you want to comment anything on, you mentioned neutrolipids and maybe cholesterols and other, did you find the right mix or was it just good luck again that you came up with that composition?
If you look at the composition for alveolar wash, you have all neutrolipids. We found, as I said, cholesterol esters, with fatty acid, 3-acylglycerol. But what is important, but we had also a little good luck, I say. We took only the phospholipid fraction to see if it is function. But we also had the other fractions we could add if we needed. But we never needed to add these neutral lipids. So that's
Thank you.
Thank you. I think this concludes